Hi, my name is Anupam Singhal, and I'm the Senior Product Manager for Antibody Discovery at Berkeley Lights. I'm going to talk to you today about how Berkeley Lights' new Opto Plasma B Discovery workflow will accelerate lead molecule discovery against difficult targets. Even the best athletes in the world know that they have to take multiple shots on target in order to hit the mark. In hockey, top scorers may need to take over 100 shots to score a goal. As a result, these athletes aim to generate high volumes of high quality shots in an effort to win every game. Drug discovery similarly requires a large number of shots on target in order to yield an effective therapeutic molecule. By some estimates, less than 5% of drug discovery campaigns successfully yield a new therapeutic. Unlike professional sports, however, this inefficiency is extremely problematic because each campaign can take over a decade and cost over $1 billion. Success rates for therapeutic antibody discovery against difficult targets has been especially poor since these antibodies often need to bind cryptic epitopes or trigger complex molecular functions. To date, only 1 out of an estimated 800 human GPCRs has an approved therapeutic antibody. To close this gap, drug developers need to find ways to increase the quantity, quality, and speed of generating lead candidates. Berkeley Lights' Opto Plasma B Discovery workflows can yield hits against difficult membrane targets like GPCRs. Genovac has successfully discovered antibodies against 3 transmembrane proteins that exemplify the common problems with difficult targets. Small extracellular regions, reducing the number of possible sites to target, high sequence homology, reducing the chances of eliciting an immune response, and poor cell surface expression, making cell-based assay screening difficult. Across all 3 targets, they found approximately 10 times more hits than traditional hybridoma campaigns. I'm now going to describe how our newest Opto Plasma B Discovery workflow accelerates the translation of these hits into lead molecules. Antibody discovery using the traditional hybridoma approach requires immortalization of B cells by cell fusion, followed by a lengthy process of expansion, subcloning, and screening. The immortalization step often results in the death of greater than 90% of B cells and therefore results in a significant loss of B cell diversity. New single B cell technologies seek to access greater B cell diversity by bypassing cell fusion. However, many of these approaches provide little to no functional information. This results in significant time, labor, and expense for sequencing, cloning, re-expressing, and characterizing irrelevant antibodies that do not bind the correct epitope or fail to demonstrate the desired function. The Opto Plasma B Discovery 4.0 workflow maximizes diversity of antibodies through direct functional profiling of plasma B cells from immunized mice. In just 1 week, the workflow enables recovery of thousands of hits by screening up to 100,000 plasma B cells, down selection of lead candidates by functional profiling, and sequencing and re-expression of over 1,000 functionally characterized antibodies. The Beacon system fully automates cell sample handling, screening, cDNA synthesis, and sequence recovery. A 96-well plate containing a purified plasma B cell sample is inserted into the Beacon. The Beacon system then automatically loads the cell sample into OptoSelect chips. Each OptoSelect chip contains over 20,000 nanopens, each less than 1 nanoliter in volume, or greater than 10,000 times smaller than a well in a 96-well plate. Plasma B cells are cloned into nanopens using Berkeley Lights' OptoSelect technology, in which cells are manipulated by light. This light-based manipulation can be performed in parallel across hundreds to thousands of cells, and tens of thousands of cells can be cloned across multiple chips in less than 4 hours. Nanopens enable rapid, precise, and highly sensitive assays. The small volumes enable secreted antibody concentrations to reach detectable levels in a matter of minutes. In the assay depicted on this slide, secreted antibodies from plasma B cells bind antigen-coated beads and are detected by binding of the secondary antibody, resulting in a fluorescent halo above pens containing antigen-specific plasma B cells. This assay is performed on tens of thousands of cells simultaneously. The video on this slide depicts a single field of view in which hundreds of cells are screened to identify dozens of hits. Antibody specificity and cross-reactivity can be assessed by performing multiple assays either sequentially or in parallel. This figure depicts 3 assays run in parallel that demonstrated simultaneous identification of antibodies that are specific to antigen X, that are cross-reactive to both antigens X and Y, as well as antibodies that are not specific to either antigen. Cell binding assays can also be used to down-select lead candidates against membrane targets. In these assays, beads are replaced with cells that are overexpressing the target antigen on the cell membrane. Antibodies specific to the cell surface target are again selected based on the fluorescent halos that form on top of pens containing antigen-specific plasma B cells. Following single plasma B cell cloning and assays to identify antigen-specific antibodies, OptoSeq barcoded BCR is used to automate sequence recovery on chip. mRNA capture beads with dual genetic and optical barcodes are loaded into specific pens, and plasma B cells are lysed to capture their mRNA. Reverse transcription is then performed on chip in order to generate stable cDNA. Following cDNA synthesis, the optical barcode for each bead is read out by fluorescence imaging. These optical barcodes are stored in software to subsequently link each antibody sequence to function. The barcoded beads are then automatically recovered in bulk. Over 1,000 hits can be recovered in a single 96-well plate, and additional hits can be recovered in multiple well plates. We demonstrated the recovery of nearly 12,000 hits from 2 Opto Plasma B Discovery workflows performed in 1 week from a single plasma B cell sample. Using a combination of fragmentation, NGS sequencing, and bioinformatics, OptoSeq barcoded BCR enables rapid and accurate sequencing of the full-length variable region of antibody genes. The ability to reconstruct full-length antibody genes from short reads enables high-throughput sequencing of over 1,000 antibodies from a single run on any Illumina sequencing platform. In the graph on this slide, over 500 paired heavy light chain antibody sequences were recovered from a single 96-well plate in a single MiSeq run. Production of lead molecules for downstream characterization typically includes costly gene synthesis and laborious bacterial cloning. OptoBCR rapid reexpression enables production of antibodies in just 1 week to confirm function in plate-based assays. Amplified cDNA is cloned into expression constructs in 1 step, enabling expression of over 1,000 antibodies for rapid functional confirmation. In this graph, over 80% of over 250 re-expressed antibodies exhibited antigen specificity in plate-based ELISA assays. The new Opto Plasma B Discovery workflow advances from B cells to lead molecules in just 1 week. The workflow enables recovery of thousands of hits by screening up to 100,000 plasma B cells, down selection of lead candidates by functional profiling, and sequencing and re-expression of over 1,000 functionally characterized antibodies. By increasing the quantity, quality, and speed of generating shots on goal, Berkeley Lights' Opto Plasma B Discovery workflow will enable drug developers to discover lead candidates against difficult targets. Thank you. Please reach out to us if you're interested in learning more.